Okay, here we go. I want to finish talking about selection on continuous traits today. And where I left off, I'd introduced the concept of reaction norms, which is a mathematical relationship between phenotype and environment. So if you raise wild type fruit flies at different temperatures, the number of facets in their compound eyes varies as a function of temperature. And you can, in fact, graph that function, which has been done here. That's that blue line. Sorry, not the blue, the black line up at the very top. And fruit flies with different genotypes, the ones called infrabar and ultrabar, those also, those genotypes have different reaction norms. And the reaction norm is the equation that those line segments describe. You could, in principle, write out a linear equation, temperature equals something times facet number plus such and such, and that would describe the norm of reaction. And in this case, different genotypes don't produce different phenotypes, they produce different reaction norms. Infrabar flies and ultrabar flies have the same phenotype if you raise them both at about 17 degrees Celsius. They have very different phenotypes if you raise both at 30 degrees Celsius. So the difference isn't the phenotype, it's the reaction norm. There's a really cool case study of this in humans, and it came out about 20 years ago. I've looked for some updates, and as far as I know, this is still, a gold standard example of this. Caspi et al. Studied several hundred human males, and there's a brain enzyme, monoamine oxidase A, that breaks down neurotransmitters, and different alleles of the gene for monoamine oxidase A differ in the promoter sequences. And the upshot is that some men have low MAOA activity and some have high enzyme activity. And this is genetically controlled. It's inherited. We know the DNA difference between high and low MAOA activity. Since that particular enzyme is involved in neurotransmitter processing, it's not unreasonable to think that this could have something to do with behavior. And Caspi et al. Scored all of the participants for antisocial behavior. The psychologists are very good at coming up with tests for all sorts of things, where they sit you down and make you answer questions, or look at ink blots, or whatever it is that they do, and they can come up with various aspects of behavior and personality and assign scores to them. So there is a standard test for antisocial behavior, measuring the probability that you're going to go shoot up a liquor store and steal the cash register, things like that. So they scored them for antisocial behavior, and they also recorded whether the men had been abused as children. So you've got three different things: the genotype of monoamine oxidase A, the degree of abuse in childhood, and the antisocial behavior test score. And they got this: men with high monoamine oxidase activity, for all men, your incidence of antisocial behavior goes up with mistreatment in childhood. Children that are abused may grow up to be abusers themselves or otherwise have severe behavioral problems. But notice the genetic difference. Men with high monoamine oxidase activity tended to be a little bit more antisocial than men with low if they grew up in homes without much abuse, but they were quite a bit less antisocial if they grow up in homes with high abuse. Men with low monoamine oxidase activity were the least antisocial if they had no abuse, but the worst if they had severe abuse. So these lines right here are in fact describing reaction norms. There is no gene for antisocial behavior as such. We do have a genetic difference that contributes to the incidence of antisocial behavior, but that gene doesn't give you a phenotype, it gives you a norm of reaction. What the gene influences is how a boy responds to the level of abuse that he receives in childhood. And this is important because it's fashionable, or at least it used to be, to look for. Sorry, I'm just checking the participants. I was about to let somebody in and then they seem to have vanished off the radar. Hang on. Okay, there we go. It used to be fashionable to look for genes for this. See if we could find the gene for antisocial behavior. About 30 years ago, there was a lot of speculation about whether there was a gay gene, a gene for homosexual orientation. You get it with physical diseases. People used to look a lot for the gene for schizophrenia or the gene for depression or the gene for this and the gene for that, even genes for cancer and things like that. There are genes that are linked to all of these, but that doesn't mean that they cause them. There may not necessarily be one genotype that guarantees you will develop schizophrenia or one genotype that guarantees that you'll develop cancer. Rather, there are genes that may give you different norms of reaction, and the environment ultimately selects what your phenotype is. This is the kind of gene cross environment variance that we've talked about, and it's all over the place. And I feel like it's important to know this as we look for the genetic background for all of these ills that plague humanity, is that it's not nearly as simple as finding one gene for one thing. And in fact, you can select for different reaction norms. This is the picture that I wanted to include Thursday. These are hydrangeas. I think this is in the floral department at Bougie Kroger, the one over on Salem. They'll have them at about this time of year. And the flower color is a function of a norm of reaction. Specifically, flower color is correlated with soil acidity. Actually, there's an intermediate step. In acid soils, there are more free aluminum ions. In alkaline soils, aluminum tends to bond with grains of clay. So there's less free aluminum in alkaline soils. And aluminum plays a role in pigment biosynthesis. If you have acid soil, the plant accumulates aluminum, and the aluminum is used, I'm not quite sure how, but it's used to make that blue pigment. In alkaline soils, the plant can't take up aluminum because the aluminum tends to stick to the clay. And what you end up with is pink flowers. But there are lots of different varieties of hydrangea. And I think my mom's got them all in her front garden. And some varieties have been selected to turn blue easily. They can make that pigment using less aluminum. Others haven't. So different varieties of hydrangea have different reaction norms, and you can actually select for this. You're not selecting for blue hydrangeas. You're selecting for hydrangeas that easily turn blue and don't need large amounts of acidity because they can make blue pigment with less aluminum. If that makes any sense, you can select for strains that have different reaction norms. Incidentally, the white hydrangeas have been produced by artificial selection, not to make any pigment at all. So you can have varieties that simply don't make anthocyanin pigments under any circumstances, and that's not a norm of reaction. That's a case where the genes for pigment have just been completely shut off, silenced, no matter what the environment is. So with all of this in mind, you can create blue, purple, pink, and white hydrangeas and sell them at about this time of year. Riffing a little bit on what I was saying about how we have to be aware that just because something has a genetic background doesn't mean there is a specific gene for it. Back in 2004, a gent named Dean Hamer published a book called The God Gene. Now, what that means is psychologists come up with tests for all kinds of personality traits, and they've identified a trait called self-transcendence. That more or less is how, quote, spiritual somebody is. There's no evidence at all that your specific religion is encoded in your genes. People have actually looked for correlations between, parental religious denomination and children's religious denomination, and it's practically zero. There's no evidence for genes that will influence you to be a Baptist or a Buddhist or a Baha'i or whatever it is. If you're a Presbyterian, it may be predestined, but not by the genes. There is a trait, self-transcendence has a heritability of about 0.4, and that isn't linked with a specific religion, but someone with high self-transcendence would be more likely to Ronnie, your mic is on. Could you mute that? Sorry, I don't mean to put you on the spot, but I was hearing some noise. The person, if you have high self-transcendence, you have a high likelihood that you'll go to religious leaders for advice, that you'll spend time praying or meditating, that you'll feel a sense of awe and wonder at the universe, that you'll seek advice from religious or spiritually important texts. People with low self-transcendence tend not to do these things. They're less likely to read spiritual texts or meditate or pray or anything like that. And self-transcendence is supposed to be independent of the specific religion. You could be a highly self-transcendent Baptist, Buddhist, Catholic, Wiccan, whatever it might be. Hamer argued that there was a correlation between self-transcendence and alleles of yet another gene involved in neurotransmitter processing, this one called VMAT2. And it makes for a really cool story, and it was a best-selling book back in its day. The problem is that VMAT2 accounts for roughly 1% of the variance in self-transcendence. It does account for some, but it accounts for a minuscule fraction. Let me show you what this looks like on the whiteboard, just for a moment. Let's say I've got a graph, and what we're measuring is number of people in a sample. And on the x-axis, we're measuring self-transcendence. And let's say we sample a whole bunch of people and we take people with one allele of VMAT2, we'd find something like this, our old friend, the bell curve, in that some of the people with one allele of VMAT2 are total God botherers, the ones over here on the right. And some of the people with VMAT2 are complete cement heads, don't have any urge to, do anything spiritual or go to church or meditate or feel awe or anything like that. And most of us schlubs are somewhere in the middle. What Hamer found was if you took a different genotype of VMAT, you got this. Okay, there's almost no difference. People with that other VMAT allele are slightly, on average, more likely to have higher self-transcendence, but there's so much overlap, and the differences in the means, are absolutely minuscule. The mean of one is right there, and the mean of the other is right there. And the distance between those means is extremely small compared to the width of the bell curves. So only the tiniest amount of variance in self-transcendence is explicable by variation at the VMAT locus. Okay, so yeah, as far as I know, no follow-up studies have found any individual genes at all with anything more than the most minor effect on human spirituality. There is a genetic effect, but it seems to be very highly epistatic, meaning it requires lots of genes. And even then, the narrow sense heritability is only about 0.4. That's not particularly high. That's much less than the narrow sense heritability for human height, for example. And there's one other thing that seems counterintuitive. Heritability is always relative to a particular human population. When you test a lot of human populations for height, you do tend to get an average of about 0.7, but that does not mean that that's the official heritability for all of humanity. Heritability can actually increase and decrease with time. And I came across an experiment in '85, very important in Arkansas, where our state bird is the Tyson chicken. It was an selection experiment trying to select chickens for increased meat production. So, this works as you would expect. They took the biggest, meatiest chickens and let them breed, and the less meaty chickens ended up in the stew. Repeat this over several generations, and for the first 4 generations, narrow sense heritability of meat production was almost 0.5. Okay, there's a fairly strong genetic component to that. After 4 generations, it started slipping. It got down to about half of what it had been, between 0.21 and 0.29. So the narrow sense heritability of meat production is not fixed for the species Gallus domesticus. Selection can actually cause it to decrease. And the reason is the more you select, the less variance you have, the less variation, because that's the point of artificial selection in the first place. You don't want to breed 2 fat, meaty chickens together and get a flock of scrawny yard birds, right? You want to breed meaty chickens together and get more meaty chickens. That's the point of selection in the first place. And the upshot is that as you select a population more and more, as you get rid of the scrawny chickens and save only the meatiest chickens, that decreases the variance in the population, right? That phenotype varies less and less. And the genetic diversity also varies less and less. And the upshot is that narrow sense heritability goes down as the variance of the entire population in every respect goes down. Those chickens start looking more and more like each other. There's less and less total phenotypic variation, and there's less genetic variation, right? As you get rid of genetic variation as you do that artificial selection. And this is why there was a study of German shepherds about 20 years ago looking at heritability of behavioral traits. And it turns out to be ridiculously low, between about 0.1 and about 0.24. Here's the thing, we know that many behaviors are strongly inherited because we've been selecting for them for many years. You can select for behavioral traits in dogs. Sheepdogs are naturals at herding. I had a professor in Berkeley who owned a Shetland sheepdog. And as far as I know, that dog had never seen sheep in its life. But when the neighborhood kids would come out to play, she'd let the dog out and the dog would run around and herd those kids, or at least it would try. I've seen breeds of other dog that wouldn't do that. We used to have a little rat terrier, and I'm pretty sure that dog wouldn't herd anything. Bark, yes. Herd it, no. We've selected for things like soft mouth in retrievers, right? The ability to retrieve a duck without shredding it. We've selected for swimming love of water in breeds like the Newfoundland, which was bred as a water retriever and a water rescue dog. We have selected for things like this, and they're part of the profile of many dog breeds. So if you look at domestic dogs as a whole, there's a great deal of variation. But when you're looking at just German shepherds, that's a population that was probably founded by a very small number of individuals. And that population at this stage is probably fairly inbred. As only the champion German shepherds in every generation are likely to pass on their genes. And, the iffy German shepherds are the ones that tend not to. So the idea is that within German shepherds, heritability of these behavioral traits is very low. What that means is not that genes have nothing to do with behavior. It's because German shepherds don't vary very much. They're not very different from each other because they descend from a small founding population. They've been subject to heavy selection. They are all pretty much like each other. And the phenotypic variance in general is very low. And the genetic variance is also very low. So these heritability measures are basically statistical noise because German shepherds don't vary much in their behaviors or in their genes. And again, that's typical of artificially selected varieties. You don't want them to vary much in their behavior or in their features, right? You don't want to mate two German shepherds together and get a Chihuahua, right? You want to mate a German shepherd with good guarding ability and get more German shepherds with good guarding ability, because that's how you make your money if you're a breeder. So the upshot of that is that when you look within a population, especially one that isn't very diverse, you may get very low heritability scores. Just like if you were to do that experiment again on chickens that have been bred for meat, you get increasingly low heritability scores because that population comes from a small number of founders and it's been subject to selection. There's not much variance at all. So heritability isn't really meaningful without reference to the population that it comes from. So even something that we know is under pretty strong genetic influence might turn out to have very low heritability when you actually measure it in a real world population. So yeah, that's crucial. A trait may have no heritability and yet be genetically inherited. German Shepherds have very little genetic variation to begin with. They also may have been selected about as far in a direction as they can go. I don't know about German Shepherd behaviors, but if you were to select chickens for increasing meat, you would eventually end up with breeds of chickens that are so heavy they can't walk. Or I'm told this is what actually does happen to meat chickens. They're so heavy that their circulatory systems give out if you try to raise them to be old. The time that they're slaughtered is about the time they would die anyway. So in that case, when you're running up against a wall and you can't select them any further in that direction, you may just have no variance and no narrow sense heritability that you can measure, and yet the trait is still under genetic control. That's a sticky point, but heritability is not the same thing as saying that a trait is under strict genetic control. Now we can get back to the bell curve. If you remember, this was the one that argued that persons of African descent, on average, have lower intelligence, lower IQ than persons of European descent. And that this is important for understanding American life and has implications for the kinds of political policy that we should be presenting. Some of this research, by the way, was actually funded by this think tank called the Pioneer Fund, which has been around since the 40s and was founded by a couple of billionaires and a couple of disgruntled scientists directly to promote white supremacy, by the way. Some of the money that is coming for research like this is pretty damn dirty. But where the funding is coming from may not matter as much as are they right or not. I mean, they do have data, but does it mean what they think it does? Stephen Jay Gould pointed out that for Murray and Herrnstein's argument to work, intelligence had to be, first of all, you have to measure it by a number. Intelligence has to be rankable in linear order. There has to be some way in which I could test a bunch of people and then line them up in a single file line from highest to lowest. Like height. Height is measurable by a single number, and you can use it to rank people. I could line you up in a line from tallest to shortest. Intelligence also has to have a genetic basis, and intelligence also has to be effectively unchangeable. It has to be something that's not particularly affected by the environment, and it's not affected by things that you might do during your life. Well, intelligence consists of so many subskills and aspects that I've seen it seriously questioned whether any single measurement can capture it all. IQ does correlate with some abilities that people have, but it sure as hell isn't everything. I've heard it said the only thing that the IQ test measures is how good you are at taking IQ tests. And I say this as someone a little sensitive about this because my sister is a professional musician. She didn't get very good grades in school, and she never thought of herself as particularly book smart. Um, some of that was due to undiagnosed ADHD. Um, and some of that is simply due to the fact that she has much greater creative intelligence than well, certainly than I do, as far as creating with sound. She's a professional musician who has worked in lots of different genres, plays a number of instruments, had one album a few years back that was actually nominated for a Grammy. Um, she is extremely intelligent. She has a different flavor of intelligence than I do. And I don't really know what our numbers are. We both had our IQs tested a couple of times in childhood, and my parents never told me. But I'm reasonably sure that whatever those numbers are, they don't reflect uh, us. They don't reflect the different ways in which we see the world and do things with information. Um, so yeah, we've just got different flavors of intelligence. Now, I got told by a psychology major who took this class a couple of years ago that psychologists do have a number called G that does seem to correlate positively with many different aspects of that intelligence. It seems to correlate with, just general brain processing power. Um, Stephen Jay Gould has actually laid into the idea of G, Spearman's G, and criticized the idea. And this is getting a little bit outside of my wheelhouse. Let's assume for now that you can use something like Spearman's G to measure something that correlates to general intelligence on some kind of overall scale, and rank that. So even if we grant points 1 and 2, where Murray and Herrnstein fall apart is because traits may have high heritability. And in fact, IQ has been estimated at having a heritability of about 0.8, as much or a little more as height. So you'd think that might wrap things up. But what you have to remember is that traits can have high heritability and still be profoundly affected by the environment. And here's a couple of stands of corn. The one on the left is a farm field in Iowa. The soil is great. The farmer has bombed the soil with fertilizer, sprayed to kill off the pests, given the corn all the fertilizer and all the water it can possibly handle, and that corn is growing like you wouldn't believe, because Iowa, one state, produces something like 10% of the world's corn. So that's an extremely lush environment. The corn plants vary in height, and we can measure the heritability of that height, and it's probably very high. On the other hand, on the right, there we've got corn growing in a much poorer environment. You can see the soil is drier. There's not nearly as much water. There's probably not nearly as much fertilizer. And the corn is growing much more low. We could go out there and measure the narrow sense heritability of height, and we'd also find that it's very high. Tall corn plants tend to have tall offspring. Short corn plants tend to have short offspring. So we'd find that there is very high heritability for corn height, and yet there's still a powerful environmental impact on corn height, and the difference between populations would disappear if environments were the same. Let me see if I can graph this. Let's go back over to the whiteboard again. And let's clear all of this. Whoops, that's not what I want. black, draw. Okay. Let's say we're measuring narrow sense heritability like we did before for height. So here we have mean parental height. And over here we have mean offspring height. In that field where we've got in that field where we've got that rich field where we've got there, where we've got corn growing very well, we might get data that looks like this. All right. And if we draw a line through that, we'd find a very strong correlation between mean parental height and mean offspring height. All right, if we went to that stunted field, the one with very little water, the one with bad soil or whatever, we'd probably get something more like this. Okay. And let's draw our regression line. We might end up with a regression line like that. Okay. See the difference? In both of these data sets, we'd have a very high narrow sense heritability, the slope of that line. In fact, the way I drew the data, the slope of the line is 1. Now, if the slope of the line is 1, if narrow sense heritability is 1, you might think the trait is under complete genetic control. And yet you can see that environment plays an extremely powerful role as well. And in fact, if we could take seed from that plant that's from those plants that are in the bad field and plant it in the good field, then we'd probably see, you could imagine that red data set sliding up and to the right and overlapping the blue data set. So you can have a trait that has, 100% narrow sense heritability, a narrow sense heritability of 1, and still have very strong environmental effects on that trait. So again, heritability is not the same thing as genetic determinism. Narrow sense heritability is the fraction of variance within a population that is due to the additive effects of genes. But when you start comparing across populations, you can find very strong environmental effects, even if there's no environmental variability within a population. Okay, this may be a little bit tricky to get around, but I think it's an important distinction to make. And I think this is the error that Murray and Herrnstein ended up making. The way to test this would be to take seeds from both the corn in the good field and the corn in the rotten field and raise them under identical conditions to do what we call a common garden experiment. And that's actually what we'd have to do in order to prove the bell curves argument. It wouldn't really be ethical to do, but if we seriously wanted to test the hypothesis that people of African descent are less intelligent than people of European descent on average, we'd have to take a bunch of babies of African descent and raise them in exactly the same environment as that experienced by people of European descent. Or do it the other way around and take a bunch of European babies and raise them under conditions that are typical of the conditions experienced by people of African descent. basically we'd have to raise a bunch of white kids in black households and black kids in white households. And it's not really ethical to do that in any kind of controlled experiment. People tend not to like it when you take babies away from their moms. Murray and Herrnstein just kind of punted the ball by claiming that environmental differences of this magnitude and pattern are implausible. They didn't actually offer any tests of that. They just kind of argued that they couldn't believe that there were big environmental differences between the environment experienced by black kids and the environment experienced by white kids. In some cases, there may not be. There are African Americans that have, middle class lifestyles as nice as what you'd see on the Cosby Family without the creep factor. And certainly there are kids of European descent that grow up in very rough conditions as well. It's not nearly that cut and dried. For the population that Murray and Herrnstein were looking at, that population might actually be old enough that things like lead paint in houses could be a factor. Lead used to be used in paint up until I want to say the 60s or the 70s. And African American neighborhoods tended to have lead paint persisting for a lot longer than others. So you had problems of low level lead poisoning that was hitting some communities more than others. And that is one thing that will knock your IQ points down quite a bit if you get lead poisoning as you're growing up. Could have also come into drinking water. If you look at the situation in Flint, Michigan, for example, where a change in water source put a lot of lead into the drinking water of a primarily working class African American town. So that's the big thing that Murray and Herrnstein asserted, but without ever testing. And it's not that easy to test, and I don't know if a proper test is ever going to be made, but it's certainly not something we can rule out. And it's something that you always have to remember when you're talking about the inheritance of complex traits, is that they can have high heritabilities and yet at the same time be very subject to environmental influences. The two are not mutually exclusive. And this kind of is the kind of thing that happens all the time. And it's almost certainly what happens in the case of intelligence. Just because intelligence does have a high heritability does not mean at all that environment cannot affect it profoundly. That, everything from better education to lower levels of lead, these are very worthy goals that are still worth pursuing. The political lesson that Murray and Herrnstein took was basically, it makes no sense in trying to spend money to improve education and housing and things like that for the Black community because they're just intrinsically stupid. They didn't quite put it in those terms, but it's there all over the damn book. And there's simply no reason whatsoever to believe that's true, nowhere close to true. So there. Okay, I tell you what, I could end this right now. That was all I had in mind for talking about, but we've still got about 20 minutes. So how about we have a word about sex? I will assume there are no objections to going on talking about sex. And I'll start with a question that I have often asked myself, especially in high school. What's the point of sex in the first place? It seemed kind of, well, never mind. I was kind of an angsty teen who did not do particularly well in high school. I'm over it now. No, really, I am. No, really, it's okay. It's just fine. Okay, right. Imagine we've got a population where there are members that reproduce sexually and members that reproduce asexually. One of the things that I know that Dr. Noyes has worked on is a particular species of flowering plant that actually varies like this. You'll find mixed populations of sexual and asexual members. There are some animals that reproduce like this as well, and I'll show you one by the time we're done. You would think that if you had a population of asexuals and a population of sexual individuals, the asexuals would take over because it takes two sexual individuals to reproduce, right? If you're a sexual individual, you have to blend your DNA with another sexual individual. Asexual individuals can just reproduce by budding or splitting or something like that. So every asexual can reproduce. Sexuals have to pair up to do it. And you would think on the face of it that if you had a population of mixed asexuals and sexuals, the asexuals would take over. They would outbreed the sexuals, and the sexual individuals would be at a disadvantage that John Maynard Smith, one of the grand old men of modern evolutionary biology, referred to as the twofold cost of sex. Sexual reproduction also carries certain costs with it. Seeking mates is energetically costly. You have to take time away from feeding and spend some time flying around and looking for a mate and maybe fighting for a mate, or growing ridiculous orange air sacs on your chest and making booming noises to find a mate, which you do if you're a prairie chicken, or all of the crazy things that animals do to search for mates. Producing gametes carries a cost with it, especially if there's something like ostrich eggs, which represent a huge expenditure of energy. And you've also got the risk that if you mingle your genes with a partner, you'll produce offspring with a less well-adapted genotype. So it would seem like sexual reproduction's at a disadvantage. And yet, when we look, it is surprisingly rare to find species that reproduce only asexually. There's a lot that switch hit between sexual and asexual stages. You victims of parasitology class this past fall might remember that that's pretty common in parasites. There's free living organisms that will do it too, like those fungus aphids that reproduce asexually so they can use a resource very, very quickly and then switch over to sex when times get hard. That's a fairly common pattern, but there's not many animal species that exclusively reproduce without sex. So sexual reproduction is, across the animal kingdom, nearly universal, very common in plants as well, although I think there are more cases of asexual plants. Turns out that sex has some benefits. One is that it breaks up something that we haven't talked about called linkage disequilibrium. Linkage disequilibrium is what you get when allele frequencies are taken out of Hardy-Weinberg equilibrium, not because they're being selected for, but because they are genetically linked to a completely different gene that is being selected on. Imagine for a moment in humans if genes for blue eye color were physically very close together on the chromosome to a gene for disgusting body odor. What we'd expect to find is that the frequency of blue eyes would go down not because blue eyes were being selected for or against, not because there was anything, badly adapted or unattractive about blue eyes, but because the blue-eyed allele was physically close on a chromosome to alleles that were being selected for because they made you smell bad. Okay, that would be linkage disequilibrium. Selection at one locus affects allele frequencies at a different locus. Sometimes this is through physical linkage, genes being located close to each other on a chromosome. There can be other effects that have the effect of creating linkage disequilibrium, even if the genes aren't physically close. That, by the way, is yet another reason why you can find allele frequencies that aren't in Hardy-Weinberg. They may be out of equilibrium, not because they are the targets of selection, but because something that they're linked to is the target of selection. And sexual reproduction breaks that up, because sexual reproduction is basically shuffling DNA. You know that genetic linkage is broken up when chromosomes cross over during meiosis. I'm assuming you all vaguely remember that there's this funky division called meiosis that produces gametes. And this is where the homologous chromosomes pair up and cross over. That crossing over is what will break up linkage disequilibrium. It will enable people to inherit blue eyes, but not stinky body odor. A Russian guy by the name of Mueller was a pioneer in studying mutations, by the way. He lived back in the early 20th century. He was the first to show that physical damage to DNA caused by radiation resulted in mutations. He's the one that made that link. And he proposed a model called Mueller's ratchet. And if you don't know, a ratchet is a wheel with jagged teeth that can only turn in one direction. The example you might be most familiar with would be a socket wrench, right? If you put the socket wrench over a bolt and you push the handle one way, you turn the socket. If you push the handle in the other direction, you don't turn the socket, right? The socket wrench just makes that rattling noise. That's a ratchet. You can turn the socket relative to the handle in one direction, but you can't turn it by pushing in the other direction. Mueller's ratchet is metaphorical. You could also imagine it as being a wheel that will only turn in one direction, but it refers to the tendency of an asexual line to accumulate more and more negative mutations. Think here about, take a picture, make a photocopy, make a copy of that copy, make a copy of the copy of the copy. Keep doing that, and within about 10 generations, you're going to have absolute sludge. And the reason is that even the best photocopier at every generation introduces a little bit of noise, a few flecks, a few distortions. At every step, at every round of photocopying, you introduce a little bit of noise. And realistically, you never remove any of that noise. So even if the photocopier is very, very good, the noise builds up from one generation to the next, and there's no way to purge it. There's no way to get rid of it. And so ultimately, after, I think 10 generations or so would be typical, you end up with a completely illegible piece of sludge on the paper. The same thing happens with asexual reproduction. DNA replication is very, very good, but it's not 100% perfect. And every time you replicate it, you'll get mutations. Some of those mutations might be neutral. Very occasionally, some of them might be beneficial. Um, some of them are going to be bad for you. And the problem is that in just asexual reproduction, you've got no way to get rid of the negative mutations. They just tend to build up over time, and eventually the lineage of organisms will fail. There's a single cell protist called paramecium that you might remember seeing in 1440, I think, maybe 1441. It's a pretty big single-celled critter that swims around and looks kind of like a slipper. They will reproduce asexually, but after about 150 generations, they start being less vigorous, they start getting shorter, and by about 200 generations, an asexual lineage of paramecium will die. They can reproduce asexually for a while, but eventually they have to reproduce sexually. And that's because sexual reproduction breaks Mueller's ratchet. In sexual reproduction, you're taking your DNA and combining it with somebody else's. That's the most basic definition of all of it. And whenever you do that, there is at least a chance that the DNA that you pass on will not be the DNA that contains the negative mutations. You might be a carrier of some negative gene, but there is at least a probability that that negative gene is not going to be one of the ones that you pass on. So you break Mueller's ratchet that way. A sexual line has the ability to purge negative mutations. The third great thing about sex is that it generates variation, because you're shuffling your DNA and mixing it up with somebody else's. And that turns out to be beneficial in itself, especially if the environment is shifting or the environment contains a lot of competitors or pathogens or parasites. Here's a guy named Lee Van Valen, not the great guitarist who died recently, an evolutionary biologist at the University of Chicago, who in the 70s came up with something he called the Red Queen hypothesis. Why is it called that, you ask? Okay, Lewis Carroll wrote Alice's Adventures in Wonderland, which I'm sure you've heard of before. And if you remember Alice in Wonderland, one of the bits is that Alice ends up in the middle of a surreal game of cards, right? There's the Queen of Hearts and the King of Hearts and the Jack of Hearts. There's that weird bit where everybody's playing croquet and the cards are making arches. This does show up in the Disney movie, as I recall. Maybe you've seen that. So Alice in Wonderland is partly based on a weird card game. The sequel is called Through the Looking Glass, and Alice ends up in a weird world that's based on a chess game. And she encounters the White Queen and the White King and the Red Queen, because this is a white and red, not a white and black chess set. And here's Alice encountering the Red Queen. You can see the Red Queen is a chess queen that sprouted feet. And it's very weird because Lewis Carroll was weird. And there's a bit where the Red Queen and Alice suddenly start running. And Alice is completely tired out, but notices that they haven't moved. And Alice says, where I'm from, when I run, I usually get somewhere. And the Red Queen says, that's stupid. Now here you see, it takes all the running you can do to stay in the same place. That's why Van Valen called this the Red Queen hypothesis. And it states that the main driving force behind evolution is ecology, ecological interactions among species, predation, competition for resources, parasitism, disease, symbiosis. All of these ecological interactions are ultimately what causes natural selection to happen. If that's the case, every adaptation in one species will force others in its ecosystem to adapt. Let's say that, let's just pretend it's the nature reserve and some insect um, has a mutation and it's useful, and the insect adapts to have more efficient jaws. Now, that insect is going to fly around and eat more plants, right? That means the plants are now under selection because they've got an insect that eats them. So the plants now have to evolve defenses. Maybe that's tougher leaves, maybe that's poisonous leaves. Let's say it's tougher leaves. Okay, now it's tougher leaves, and so the insect is under selective pressure to evolve tougher mouths. Okay, on the other hand, maybe the tougher leaves, plants that use tougher leaves now have less energy for something else. So maybe tougher leaves means there's less energy for seeds. So now the plants are under selective pressure to generate more energy to produce seeds. Okay, now that causes something else. And the point is that every species in that ecosystem is being subjected to selective pressure every time every other species changes. And so every species constantly has to adapt just to hang on to its ecological niche. A species constantly has to change just to keep in line with its competitors, its parasites, its predators, and its pathogens, and everything else. What this has to do with sex is that sex is how you maintain the capacity for change. You maintain variation in a sexual population, and that variation is the raw material for evolution to happen in the first place. So that's what the Red Queen has to do with sex. And there was a neat test of it about 25 odd years ago in New Zealand. They looked at a freshwater snail called Potamopygus antipodarum. And in this population, sexuals and asexuals can coexist in the same population. On the left, that's a map of New Zealand, and what look like hat pins sticking into New Zealand is because this is a voodoo doll made from somebody who hated that country. Actually, I'm kidding. The heads of what look like hat pins are actually tiny little pie charts. What they're measuring is the frequency of males. You can see there are some populations that are all female because the head of that pin is completely blue. There's others that have more white, and those are populations that have a greater frequency of males, up to about 0.4. So there are some populations that are entirely asexually reproducing females, and some that are sexually reproducing male and females. And the incidence of sexual individuals is directly correlated with the parasite load. Snails get infected by parasites, in particular flukes, a particular group of flatworms. And we learned this in agonizing detail in parasitology. Some of you veterans of this might remember. And the reason this matters is that when flatworms parasitize snails, they are known to destroy their gonads. It's common enough, it's got a name. It's called parasitic castration. And if you cannot reproduce because the parasites have eaten your gonads, you might as well be dead, at least as far as the evolution of the population goes. In populations where there are no parasites or very few, then the population doesn't need to be sexual so much. Well, I don't mean need, but the asexuals have the advantage. They can reproduce faster than the sexuals can. The problem is that if in an asexual population, everybody's alike, and if a parasite invades an asexual population, if it can parasitize one, it can parasitize them all. It would be like hackers attacking all of our computers if we all had the same password and never changed it, right? If everybody in this class had a password of password and we never changed it for years on end, then anybody that broke into one of our computers could break into everybody's. On the other hand, if our passwords were variable, if we all have random passwords and we keep changing them every week, then it's a lot harder for a hacker to get all of us. Well, if our passwords are variable, that's be analogous to sexual reproduction, creating genetic variation. Sex creates genetic variability, and that variability itself is useful in populations with a high parasite load, because at least some of those individuals are going to vary in ways that enable them to resist the parasite. So this seems to support the Red Queen hypothesis. And some more recent work on the snail shows that asexuals accumulate mutations at a more rapid rate, which seems to support Mueller's ratchet. Sexual organisms can get rid of deleterious mutations in ways that asexuals can't. This is work by Maureen Nyman, whose lab has been practicing asexual harassment for quite some time. Right. Okay. I'm going to go ahead and end there, and we'll talk about Darwinian demons later. Of course, there's people in the state that already think that Darwinian evolution has demons in it, but we'll get to that later, and I'll go ahead and stop recording.